So now that we've seen some evidence that the circannual rhythms appear to have some endogenous properties, at least they free run. They free run it slightly different than the 12-month period. Let's take a look at some other interesting data. As scientists were trying to figure out, as they did with the circadian rhythm, where was the master clock? Where could it maybe be? And so, not just the data that I've shown you, but plenty of other data, even beyond birds, show that in mammals and birds, plenty of evidence that under constant conditions, there's a whole host of behavioral and physiological circannual rhythms. One clue that was known early on was that starlings show circannual rhythms when they're housed in 12 hours of light and 12 hours of dark, but they don't show the circannual rhythm under slightly different photoperiods of slightly longer or slightly lower or shorter light periods. So 11 hours of light versus 13 hours of dark, or 13 hours of light and 11 hours of dark. These photo regimes, we end up with a no circannual rhythm in testicular width, like you do in 12-12. And interestingly, in the relatively shorter photo periods, we see that starlings maintained a larger testicular width compared to birds that are under longer light periods, longer than 12 hours. They, right after the initial testicular growth, they recess and they never grow back again. And so, what are some of the ideas that people came up with for where could the what is the mechanism what's generating the circannual rhythm? In ground squirrels, it was tested. There's this idea that it sort of requires a specific sequence of interrelated events, like physiological events. And if you were to disrupt one of those, you would, like taking a spring out of the machine or a gear out of the machine, it would disrupt the whole thing. And it requires all these interrelated events at certain times for the circannual rhythm to process, like a chain reaction. So interrupting one would stop that chain reaction. And so here's an example of an experiment done in the 80s where they tried to do that, taking a look here at circannual body weight fluctuations in ground squirrels. And the E and F on the bottom show controls that aren't manipulated at all, and you can see they wax and wane in their body mass over the course of the year. And experimentally, they just removed fat from the ground squirrels surgically. And of course, had controls in which they didn't remove fat. And you can see that precipitous initial dip from the surgery itself, from removing the fat. But ignoring that initial little dip, the contours of the circannual rhythm still persists. When you remove fat during a period where you would expect them to start decline in body weight, they continue to decline in body weight. They sort of have this initial recover, but they recover back to not the original mass, but a mass of what you would predict if they were on this programmed downward weight loss decline, and vice versa if they remove fat earlier in the year during this period when you expect to see ground squirrels slowly gaining mass. If you remove fat tissue after a brief period of recovery, they continue to increase. So this doesn't support this hypothesis that if you just disrupt one of these physiological processes, you'll scuttle the whole circadian rhythm. after a brief period of recovery, they continue on the same um, circannual rhythm of body mass. Another idea that came about was called the frequency demultiplication hypothesis, and it was thought that the circannual rhythm was sort of an extension of the circadian rhythm, whereas our body endogenously kept track of 24 hours over and over and over. It would keep track of days for the year, and it sort of kept track of that by like how many circadian rhythms it had cycled through, and it would sort of like reset once it reached the year threshold, and then it would start counting again. And so you could easily test that, right? You could entrain animals to a day that's greater than or less than 24 hours. And if it's simply counting how many circadian cycles it goes through until it reaches a year, if you shorten their circadian rhythm, then they should show a shorter circannual rhythm. Or if you extend it to, say, 25 hours, then you should expect to see a longer circannual rhythm. But again, doing that didn't show, didn't see any evidence of that in ground squirrels that were entrained at different T cycles. And they showed this approximately the same ranges of circannual of activity and body mass and estrus. It didn't make a difference if you entrained them to different day lengths. This was a similar experiment done in warblers as well, and trained to a 22-hour day or 26-hour day. This did not result in the predicted proportional change in circannual period. They maintained circannual periods similar to each other, despite the fact that they were on very different circadian clocks, and trained to different circadian times. So, where is the circannual clock? How would you go about locating it? Well, no surprise that the people that were interested in trying to find the physiological basis for it followed the same path as those that were interested in the circadian rhythm, just ablating different parts of the brain to see if it would disappear. And so, scientist Lesion the paraventricular nucleus, the PVN, in an attempt to alter the body mass rhythm, that had no effect. Pinealectomy, taking out the pineal gland, shortened period length in ground squirrels. So they showed like a more rapid circannual rhythm, much less than 12 months, but the rhythm still persisted. They lesioned the SCN. Maybe if it's done the circadian stuff and it's linked, maybe that's also where the circannual rhythm is. Some squirrels lost circannual rhythms, but others did not. Others whose SCN was completely gone, they still maintained a circannual rhythm. And here's the data, some of the data from that experiment. We can see those intact individuals on the lower left showing clear circannual behavior of activity. Upper left, those that had their SCN ablated, still showing circannual rhythms, but others whose SCN was ablated, they became arrhythmic. There's some mixed results here, so something's going on. It can't be just the SCN, right? Because some individuals still show the rhythm, the endogenous rhythm. Further experiments, the ventromedial, the anterior and lateral hypothalamic nuclei, no effect. Lesions to the medial preoptic nucleus, olfactory bulbs, the septal area, no effect. Removal of the gonads, no effect. One keynote or flagship set of experiments started people thinking the importance of the thyroid hormones in at least being linked to the endogenous circannual rhythm. And so this was done in sheep and also later in deer. Removal of the thyroid gland disrupted normal circannual rhythms in these animals. And so here we're taking a look at sheep, and we're measuring luteinizing hormone, which is a hormone part of the gonadal, the hypothalamic pituitary gonadal axis. So the main endocrine axis that regulates reproductive physiology and by extension behavior in vertebrates. And this course is in sheep. And we can see the THX, that means a thyroidectomy. So they're taking out the thyroid in October or December. It doesn't matter when they took it out. We see that these individuals don't show the regular decline in luteinizing hormone in an intact sheep. So they seem to lose the circadian rhythm, at least looking at it in a 1-year period. And so they thought that it must have something to do with thyroxine, which is one of the hormones that the thyroid produces. And so indeed, by giving these animals thyroxine during winter and early spring, they recover this decrease in luteinizing hormone that mirrors an intact sheep. Though the study was sort of, they had a lot of mixed results. And they discovered, because some sheep showed, when they give them these thyroxine, some sheep, like in the top graph here, show a decrease in luteinizing hormone when you would expect. But others did not, even when they gave them these thyroid hormones. And what they discovered over a series of experiments, that it had to do with subtle differences in the timing of their method of giving the thyroid hormone, thyroxine. They gave these little miniature osmotic pumps, which are just a passive way of giving sort of a chronic exposure to a hormone or any chemical. It's sort of like it's a little pill-shaped type of apparatus with the hormone inside, and it's designed in such a way to slowly dispense it over time just through osmotic pressure. And it would do so for about a week, but it doesn't do it evenly. If you take a look at the dotted lines here, this is the fluctuation in the hormone being dispensed, right? And so they have to give them 2 different pumps at the end of December and the end of January, because by the end of January it's sort of run out, it sort of reaches a peak and then declines, and so it's not a smooth administration of hormone over this 2-month period, but it dips. And what they found out was that there was a correlation between those sheep that experienced a decrease in luteinizing hormone had this window, this transition from January to February, that was better covered by exposure to thyroxine compared to ones that continue to show arrhythmic circannual hormone profile. And indeed, the individuals that they gave these mini osmotic pumps to, just one during either late December or late January, had no effect on restoring their circannual hormone profiles. And that's because administered in this fashion, they weren't getting significant levels of thyroxine during this critical window of the transition between January and February. Those individuals that they gave the pumps to, but also supplemented with sub-Q injections of thyroxine during the transitions, so you wouldn't get these 2 humps, but a more constant, even administration of hormone across the whole 2-month period. They all had a restored circannual hormone profile. And here's another example of an individual. They got got hormone mini pumps implanted in such a way to ensure that it had peak thyroxine during late January and early February, and then sub-Q injections during the same period over a 2-period process, and they were able to keep this maintained circannual rhythm. So clearly in sheep, and then in later experiments, deer, thyroxine is an important part of this mechanism of maintaining circannual rhythmicity in at least this one physiological process of circannual luteinizing hormone, which again is an important component of the estrus cycle, the reproductive cycle in these animals, helping them gear up and maintain reproductive physiological. Now, to fully understand and appreciate the endogenous circadian rhythm, we need to fully understand and appreciate photoperiodism, which is the environmental, the photoperiod is the environmental cue that regulates the circannual rhythm. It's the Zeitgeber, much like day length is the Zeitgeber for the circadian rhythm. Changes in day length over the course of the years is the Zeitgeber for the circannual rhythm. So what is photoperiodism? What is it significant for the regulation of this annual biological cycle? So we became, at least as a sort of collective Western scientific knowledge of photoperiodism in 1920 with a series of experiments done by Gardner and Allard. They first discovered the importance of day length in the regulation of a flowering tobacco plant. And plants, most tobacco strains flower in August. They noted that there was a mutant strain, Maryland mammoth, that flowered later than other tobacco plants, not until December, even when they kept it in relatively constant conditions in the greenhouse. It had much larger leaves and was taller, which are commercially desirable characteristics for tobacco plants. All right, so this was driven by ultimately by capitalistic commercial drivers, right? A bigger and better plant that can be sold. And so the Maryland mammoth was to be used in a breeding program, to produce these great tobacco plants with big leaves. You get more tobacco from one plant. If it were to be used, it would have to flower in the field when the other plants were flowering. So they would have to figure out how to flower in August instead of December. So that was their task. That's what they were trying to do. And so they tested for the effects of different levels of moisture, temperature, nutrients, and light on the flowering of this plant. All kinds of things in the greenhouse. The temperature, different types of moisture, different nutrients, different water levels, all kinds of stuff. And it didn't do anything. And it was only when they thought that why not vary the day length when thinking, well, what's the difference between August and December? Right? One of the first things, they tried was temperature. It seems like an obvious. It's colder, so maybe just cold temperature stimulates the flowering. And they eventually got to day length. And they found that was the environmental cue that influenced flowering. They moved pots of Maryland mammoth into a light, tight greenhouse every afternoon. I guess that's kind of an oxymoron, light, tight greenhouse. A light, tight structure in the greenhouse every afternoon at 4 p.m. and then back outdoors again at 9 a.m. the following morning to give it an artificial light regime of longer nights, of 17 hours to be exact, of darkness, and relatively short days of 7 hours of daylight. And it flowered. The result was flowers and seeds in July. Meanwhile, out in the field, Maryland mammoth was the ones that they weren't manipulating, was still basking in 14-hour summer days and was nowhere near to flowering. So they found the key. It's day length. And in further experiments, they found that some plants like tobacco, soybeans, they flowered in response to day lengths that were shorter than some kind of maximum. But other plants included many grasses and cereal grains. They flower in response to day lengths that were longer than some minimum. And so that's given rise today. The most commercial crops, agricultural crops, can be categorized as short day plants or long day plants, depending on when they flower and exposed to short day or long day. An example of this is marijuana. You see pictured here on the right. Most of the commercial strains now, the commercial strains of marijuana that are grown to be brought to market, and in the past also when they were being grown illegally, and still grown illegally in many states, and I guess technically still grown illegally according to federal laws, but not according to many state laws, including here in Vermont. They would grow them under short day conditions because cannabis is a short day plant, and so in order to get it to flower, that's why people grow marijuana, is to get it to flower, because that's where you have the high concentrations of trichomes, where the THC is. You have to expose it to short days. And so Garner and Allard were the first to show that the time of flowering could be advanced by exposure to long or short days, depending on this species. So the discovery of the importance of photoperiodism was first done in plants. And that term specifically was coined by a USDA scientist named Cook, but the term was initially introduced to the literature by Garner, popularized by Cook, sort of set in stone by Cook. And it's very simply the biological response of organisms to seasonal changes in day length. The original experiments done by Garner and Allard over their years of work estimated to be about $10,000 in overall cost, which is arguably one of the most sort of like best scientific investments of all time, given the billions of dollars that have been made using this simple bit of information of how to control when plants flower, which for lots and lots of plants is necessary to develop the parts of the plants that are commercially viable. Right? A lot of the things that we eat are, ripe ovums, mature seeds, for example. And so it's really has been $1 billion, maybe even $1 trillion idea in farming and horticulture, plant breed development, and so forth.